So why is that an issue and what is that? Let's start with the definition of it.
GMO, genetically modified organism, is an organism that has added its genetic material altered to have a better or our favorable traits. Today, I'm going to talk about genetically modified crops.
There are a few ways to make genetically modified crops, but the most common way is Agrobacteria Mediated Transformation.
This is the structure of Agrobacteria. Agrobacteria is a soil organism that makes tumor on a plant by passing its tDNA in its plasmid called Ti plasmid to a plant cell. By discovering this phenomenon, scientists got an idea that it might be possible for them to transplant a gene that is encoded for their favorable traits instead of tumor-inducing gene. Now let me explain the process of it. Ti plasmid is where all the important factors for the transferation of tDNA to a plant cell are located. But Ti plasmid is too big to be used for genetic engineering, so scientists made a smaller artificial plasmid and cloned tDNA region to it. And they also put two origins of replication, one for E. coli and one for Agrobacteria. Now, we have to isolate a sequence of genes that you're interested in from other organisms.
Can we use all the genes from all the organisms? Theoretically, yes. Because all the organisms on Earth have same base of DNA and same way to express it. But as species evolve differently,
While some genes are likely to be expressed in certain organisms, some genes are not. Now let's look at tDNA closer. There are genes called cytokinin, auxin, and opin which are in charge with making tumors on a plant, so remove these and insert the isolated gene. Then insert a small plasmid to E. coli that is lack of its own plasmid. Then the small plasmid will clone itself as the proliferation of E. coli takes place. Now move small plasmid back to Agrobacteria. Then Agrobacteria will look like this. It contains two plasmid, one that contains var region and one that contains tDNA. Make a wound on a tissue of the plant where it can differentiate well, such as hypocotyl and cotyledon. Then culture with solution that contains auxin and cytokinin, which promotes the cell division.
now it's var region time!
The proteins that are made as var region express detect the wound of the plant cell and make a way for tDNA to be transplanted. If the genes are well expressed, it becomes GMO. Okay, let me put it like this. Imagine you love sweet ice cream, but at some point it got salty, and you found out that your sister has been adding salt. When you found out, you switched the bag of salt to a bag of sugar. Now you have ice cream that is even sweeter. 10 genetically modified crops that are edible are approved in US, and 85% of daily food in US contains GMOs. It has numerous advantages such as increased crop yields and immunization to harmful insects. However, there is a report that mice that consume GMOs consistently die 2 to 3 times faster than those who did not consume GMOs. So it is still debatable if GMOs are safe to consume or not. So we should educate ourselves about GMOs to weigh the pros and cons. Thank you everyone for watching.